This is actually my very first visit to Hong Kong University. I've been to many universities in the world, but somehow haven't been here until now. So that's a special joy and privilege to be with you this evening. I would also like to thank this lady up here. This is Nell Whiteway, who was on our Faraday staff for several years. And Nell did a lot of the literature research, which was the background really to the talk this evening. In fact, it was the background to this particular book, which is coming out hopefully in a few weeks' time. I'm unfortunately not quite in time for the lecture this evening. So what I'm going to do this evening is to give you an abstract, really, of what's in this book, okay, which also started life as the Gifford Lectures in St Andrews University up in Scotland in the year 2012. So this lecture itself has been evolving along the way. Okay, so are we slaves to our genes? Well, if you go back in history, of course, and you look at the period roughly from 1880 to 1940, then what you find is a very powerful narrative of genetic determinism. It was believed that heredity determined race and class and mental health and intelligence. And I'm sure you are familiar with the eugenic legislation that was quite dominant in many countries in the early part of the 20th century. And you found in what we now call very sort of progressive countries, in countries, well, the states of California and countries like Denmark and Sweden and Germany and so on, you find this eugenic legislation coming in quite powerfully in the early decades of the 20th century. This quotation actually is from a very well-respected professor of zoology at his time. He was president of the New York Zoological Society, and he once declared, and I quote, that mistaken regard for what are believed to be divine laws and a sentimental belief in the sanctity of human life tend to prevent both the elimination of defective infants and the sterilization of such adults as are themselves of no value to the community. The laws of nature require the obliteration of the unfit, and human life is valuable only when it is of use to the community or race, close quote. Okay, this is a respected American professor in mainstream science in the year 1916. And of course, we all know, I think, where that eugenic legislation led. It led to the terrible laws which were passed by the Third Reich in the 1930s. In fact, they took quite a lot of their eugenic legislation from the state of California, where that was the leading state in the eugenic movement, actually, in the United States before the Second World War. And of course, the most extreme examples and applications of eugenics led to Auschwitz and Buchenwald and that terrible history that we know. Now, of course, today, such attitudes, such practices are viewed with great horror. Surely the kind of genetic determinism that nurtured eugenics is a thing of the past. Well, this evening I want to say both yes and no, I think, to that particular comment. I suspect that today's genetic determinism is more of a kind of backdoor, insidious, culturally influenced kind of determinism, maybe more absorbed by a process of osmosis, if you like, from the media, by the abuse of genetic language in daily speech, and unfortunately also from the comments of some scientists as well. This is a comment from Nature, of course, a very well-respected journal, and where you have a News and Views report a few years ago saying that, and I quote, an increasing number of studies suggest that biology can exert a significant influence on political beliefs and behaviors. And this article suggests that genes could exert a pull on attitudes concerning topics such as abortion, immigration, the death penalty, and pacifism. And this Professor John Hibbing from the University of Nebraska-Lincoln in the States is quoted in the same article as saying it is difficult to change someone's mind about political issues because their reactions are rooted in their physiology. What I find interesting and somewhat depressing in this article is the kind of assumption of determinism here. So genes and physiology are seen as somehow something different from us and our mind, and they seem to be controlling us so that we can't even change our minds. That's the kind of narrative that one sees here. And I don't know how it is here, but certainly in my country, the phrase "it's in his or her DNA" has come into daily language, trying to indicate that something is essential in that person or that organization, and probably with the implication that it cannot actually be changed. So here's Brad Pitt, whilst discussing US gun control, suggesting that it's in Americans' DNA to own a gun. I won't comment on that, okay? So, but here's the cloud computing service provider Oxygen, who assures us that our security is in their DNA. I even came across this one, I think it was the time of the Olympic Games, which claims that London has apparently had 2,000 years of urban DNA. I have no idea what that means, actually, but I think the indication is similar, that it's something essential to London and so forth. And the situation is not really helped by the insistence of the media to keep reporting the discovery of a gene for this, that, or the other. There are mean genes, gluttony genes, gangster genes, liberal genes, religious genes. I even read in the Guardian newspaper the whimsical suggestion of a geneticism gene that influences certain people to believe that behavior is influenced or caused by genes. Okay, so it's probably good to remember, and I realize this is a very interdisciplinary audience this evening, but it's good to remember that a gene is simply a stretch of DNA or RNA indeed that encodes information, and that is it. There is certainly no single gene that encodes any kind of behavior in any living organism, let alone human beings. So does it really matter then if people start thinking that their decisions are influenced and determined by their genetic endowment? I mean, what's really at stake here in this discussion? Well, I suppose we're not truly responsible for our actions, and clearly, how can we be held account for them? What happens to the justice system? What happens to our concept of love in our personal relationships? What happens to that moral responsibility that is the glue that keeps all societies together, and we lose that glue at our peril? Now, of course, today everybody knows that genes and environment together collaborate somehow or interact somehow to generate living organisms. No one seriously thinks that our daily decisions are somehow determined by our genes. I don't think anyone believes that. Everyone knows that we are the complex output product of a complex gene-environment interaction. But I think in that dichotomous language we have a problem, because the picture you can so easily get from that language is of two powerful forces that are somehow dragging human beings in different directions, so that we are somehow fragmented rather than unified persons. And so easily the impression can be given that human personhood is some sort of battleground of these competing forces. On one side we have genes, on the other side we have the environment, and somehow they're going to fight over the human personality for control. Now, if you want to go back, certainly in English, I'd be interested to know how this works actually in Cantonese or Chinese. But if you go in English, certainly, and look for the language of nature and nurture, it really goes back to John Galton, who was the first cousin of Charles Darwin, and he first introduced the contrasting terms nature and nurture in his book Of Men of Science, Then Nature and Nurture, 1874. And he emphasized, and I quote from him in this book, that no carefulness of nurture can overcome the evil tendencies of intrinsically bad physique, weak brain, or brutal disposition. Okay, he's speaking in late Victorian English here. But the point here is, and it comes through clearly in his writings, that he believed that nature was always dominant. He believed in the environmental influence, but nature always won in the end. And he referred to the phrase nature and nurture as a convenient jingle of words. It's very interesting how ever since that time, we're talking, what, 150 years ago or so, 140 years, that this has somehow framed this discussion. And it's quite an interesting experiment, which I did the other day. I put in nature plus nurture into Amazon just to see what books there might be, you know, which are having this title. And here we are. We have titles such as Nature or Nurture, Nature and Nurture, The Nurture of Nature, Nurture by Nature, Nurture through Nature, The Nature-Nurture Debates, Bridging the Gap, Nature or Nurture, The Great Brain Debate, and so on. Okay, so you can see that Galton's jingle of words, jingly phrase, has been very, very influential. And indeed, it does influence the discussion to the present day. I actually feel rather sorry for authors or publishers who try and come up with a different combination of the words nature and nurture. I think probably they've used all the possibilities up there. My point here is I've read a number of these books, by the way. I haven't read them all, and several of them are very good. I'm not trying to dismiss the value of this particular phraseology as a way of introducing the topic of behavioural genetics and so on. But I want to say this evening, I don't think really it's the most helpful. I don't think it's the most helpful in the way of understanding contemporary biology, and also it introduces this idea of dichotomy, which I would like to get us away from. In fact, the media will often talk about the nature versus nurture debate, as if there's some sort of debate, struggle going on, between these two different forces and so on. So what I want to do this evening, I want to move the goalposts, if you like. I want to change the framing of this discussion, and I want to introduce language and ideas which I think are much more faithful to where we are with the science, and also which I think interact more positively with theology, as we'll see near the end of this lecture. So here's a framework I want to suggest. It's not nearly as jingly as the one introduced by Francis Galton. It's actually the acronym DICE. I'm going to pronounce it DICE, which happens to be Latin for to be said, which is not why I chose it. But it stands for, 4 words, a bit of a mouthful, developmental integrated complementary interactionism. I said it wasn't as jingly as Dalton, okay. You can just call it dicey, okay. But the point about here, we've got 4 words here, which, if you like, are like pegs upon which you can hang 4 really important insights into contemporary biology and the way the system works to produce living organisms and people like ourselves. Now, I realize we have an interdisciplinary audience here this evening, so please forgive me, medics and biologists, because you will, I'm sure, know very well the next few minutes. But I wanted to introduce and to illustrate the concepts in DICE just by thinking about human development, just for the next few minutes. Let's start with a newly fertilized human egg, a zygote, and let's think about inheritance. Well, what we inherit from our parents, of course, is not DNA. It's not naked DNA without which by itself could do nothing. But it's a very complicated system. If you look at the mother's egg just before fertilization, it's got at least 3,000 different proteins in it. It's got at least 7,500 different messenger RNA molecules. It's got whole thousands of non-coding RNA molecules which regulate gene switching and so on. In other words, we are inheriting a very, very complicated system. And if you just had DNA, naked DNA, it would be as useless as, I don't know, some software program on a CD or something that you didn't have any computer to run on, okay? It couldn't do anything at all. And right from the beginning, there are these 2 types of environment. You've got the immediate environment of the cell, and it's all this complex molecular machinery. You've got the wider environment of the mother, which of course includes the fallopian tubule down which that zygote soon starts to travel. And you've got the mother's own broader environment. And so the DNA and the microenvironment and the macroenvironment, they're all completely integrated from the very moment of fertilization. And of course, a key part of the microenvironment of a gene is other genes, because genes behave according to the context in which they find themselves. And a rather recent finding, there's even more recent, this slide is already getting out of date, but the point is made that at least 20% of DNA, human DNA, is involved in regulation. There's a discussion about exactly, maybe more than that, it may be a bit less, but it's around, that certainly is a minimal figure probably. And it's, of course, involved in regulating how you switch on and off these 21,000 protein encoding genes, and also the 16,000 RNA genes, thereabouts, that have been described so far. And so it's these hundreds of protein transcription factors that are involved in switching all these various genes on and off. And in the early zygote, it's not the DNA which causes development to begin, it's actually the protein transcription factors inherited in the mother's egg which starts the whole process going and starts that process of switching the genes on and off. So if you like, the proteins are kind of players in the DNA orchestra that cause the genes to play this integrated symphony of life, if I can use that kind of metaphor. You need all the instruments in the orchestra to play correctly at the right moment, exactly the right moment for the correct things to happen. So then following implantation, the fetus is now exposed to a whole new array of influences coming from the macroenvironment, from the mother's blood, of course, circulating through the placenta. So you've got oxygen, you've got hormones, you've got glucose, you've got lipids and alcohol, maybe drugs if this person is taking drugs, and so on and so forth. And by the third trimester, that developing child can taste to some degree, can smell to some degree, and hear certain stimuli inside the womb in ways that do affect postnatal behavior to some degree. And then suddenly at birth, you have this huge bombardment of a whole new array of sights and smells and sounds and cats and dogs. And I don't know how we got through this, really. When I think about it, it makes me ill just to think about coming into the world. But we all have been there. And suddenly you get this massive environmental inputs going on into the newborn. It's good to remember that the infant brain is not some miniature version of the adult brain, but it's rather a self-organizing system that only self-assembles correctly when the right inputs come in at precisely the right time. So if we jump and think for a moment about the adult brain, well, adult brains contain something like 10 to the 11 neurons, but in practice twice as many as that number are created during the fetal development, and then half of the neurons are sort of pruned away during the development of the growing brain postnatally. And so in the adult brain, each neuron receives on average something like 5,000 synaptic connections. The range is actually very broad. We're talking about averages here. Some neurons actually connect up to other neurons by as many as 200,000 synapses. And so since there are 100 billion neurons in the brain, then this implies the brain contains a staggering 500 trillion synapses, clearly making the human brain the most complex known entity in the universe, as far as we have seen so far. And all of that is constructed really during the early postnatal world. First couple of years really, you've got the synaptic architecture being generated. In fact, in the fetal brain, and indeed just a day postnatally, the neuroblasts, that you don't really have mature neurons in the brain though, neuroblasts, they're still yet to go through their maturation phase. They've still got to get myelin around the sheath. They've still got to make all those synaptic connections and so on. And if you look at the increase in synaptic density in the newborn, it's amazing. I mean, you go from something like 300 grams in the newborn brain up to 900 grams in the first year of life. This just indicates the synaptic density in the first couple of years of life in the postnatal development. And you can just see this huge increase in the synaptic structure going on. So as I say, the brain is self-assembling. It's putting itself together. And there are certain critical periods at which that sensory input is really vital for the growth of specific brain areas. So if a baby is blind from birth, for example, as I'm sure you know, then the visual cortex around the back here doesn't develop properly, and you can get permanent visual impairment if that is not corrected. So for example, children born with cataracts in both eyes have large potential deficits in motion processing and just being able to move around and keep the balance and so forth. But if you have children who develop cataracts between the period of 6 and 12 months, then they have no such deficit or a much better chance of normal processing of motion and so forth. So you've got a brain there which is quite plastic in many ways, displays plasticity, especially during these early years of brain development and so forth. And that plasticity, of course, gets less as we move into adulthood. And as we go into adulthood, then in some ways this integrated interactionism between the genes, the microenvironment, the macroenvironment becomes even more striking, because there you have an adult who has free will and who makes choices about their lifestyle, and they can choose to live in lonely environments, or they can choose to live in social environments, they can choose to smoke or not, they can choose to take a lot of alcohol or not, and so on. They can choose to take a lot of exercise or to sit watching television most of their life, and so on. So free will really comes into it big time. And of course, all these factors and many more feed back into DNA regulation through the process that we call epigenetics. So epigenetics simply means all those chemical modifications of the DNA or the proteins surrounding the DNA that are involved in switching genes on and off. So that process is going on all the time. If you like, if genetics provides if the genes provides the hardware of inheritance, and epigenetics provides the software that day by day, hour by hour, minute by minute control of what genes are expressed and so on. Certainly people suffering from jet lag, just as I am. I flew in from London last night, so I would be in that cohort. Definitely my epigenome has changed between coming from London to Hong Kong. Many people have studied the epigenome following long journeys in aeroplanes, and that for sure changes your epigenome. And of course, having a big chocolate cake changes it as well, and many other things, as I've just been experimenting beforehand. But the point here is that our epigenome is under constant dynamic control all the time, and it's a way in which the environment feeds into our genes. So we should never think of a linear pathway going, from DNA information out of the cell and so on. It's always a dynamic two-way process whereby our DNA is being regulated all the time. So that's the idea in Dicey, if you like. We see how the development of the organism is the key. It's an ongoing process that continues until death. All the components of the system are tightly integrated. There's a continual interaction between all the various components of the genomic system, the microenvironment, the macroenvironment. And there are many complementary processes going on at the same time, and you need a whole lot of different disciplines to really study those complementary processes with their different techniques and different languages and so on in order to really seek to understand this complex system. So that's what I say, that I think we've now sort of bypassed the nature-nurture dichotomy. We're into something new. We're talking about complex developmental systems, and that is really what human beings is about. So I like to say that the 100% of a complex, that the phenotype of any complex organism involves genetics, and 100% involves the environment. Because that makes the point that it's interactionism all the way through that is really important. Now, I hope you'll forgive me. We've talked a lot about chocolate already this evening. So, but I know that some people who are not biologists here this evening might need something a little bit simpler. So we could just take a cake. Okay, this is a Black Forest cake. This is a highly complex system, actually, although not as complex as human beings. Okay, so most of the cake is flour. Actually, it's about 50% of the cake is flour. So if you don't have flour in that recipe, then you won't get your cake. That's definitely the case. Less than 1% is baking powder, but if you don't put in the baking powder, you definitely will not get a cake. And if you don't have the correct oven heat, then you're not going to get a Black Forest cake. You might get something else, but it won't look like that. Okay, you've got to have it just right. So the cake then is the product of a highly complex developmental process in which the proportions of the recipe are really critical for the final product, but you can't really then go back and say, well, we'll give proportionality of importance to those different components because they've been already chemically transformed by the time you get to your final cake. The baking powder, for example, no longer exists in that cake. It's been chemically transformed into something different. So this cake has emergent properties, you would say in philosophical language, which cannot be reduced. It's irreducibly complex, actually, to its components because it's the product of all these various interactions going on over a period of some hours in the oven. Okay, well, that's just something to think about if you're baking cakes or so on. Now, given this sort of account then in developmental biology, we might well want to ask, why is this dichotomous language so powerful? Why is it we still think about these sort of genes, environment, as if they come essence or nature and nurture, whatever it might be? And I think a powerful contributing factor to this comes from the world of behavioural genetics. Now, if you take anybody in the world or anybody in this room, it doesn't matter where you're from in the world, we all vary from each other by something like 0.5% of our DNA. It's actually more than a lot of people thought. Until recently, until now, we have thousands and thousands of whole genome sequences for many, many people. And of course, the variation comes from different types of mutation, types of difference in our DNA. What I just want to focus on for a moment is that which comes from what are called SNPs, single nucleotide polymorphisms, which occur in about 1 out of 1,000. They're different in about 1 out of 1,000 of all of us here in this room. As I say, it doesn't matter where we come from. And these are just single genetic letters that vary between us all. In fact, we now know that each newborn probably has around 60 on average, new mutations in their DNA which are not found in either their mother or their father, which is also quite a bit higher, I guess, than we realized before human genome sequencing came along. By the way, it depends on the age of the father, how many mutations your newborn will have, and not on the age of the mother for biological reasons. Okay, so the question then is, here's all this difference. So what difference does that make, if any, to the variation in human behaviors? And of course, that is the study known as quantitative behavioural genetics. And there are quite a number of books there and quite a number of useful textbooks and so on. And we're going to just touch on it briefly this evening. And of course, the aim is to find out, as I say, whether that variation really makes any difference, and how would you know? Well, what quantitative behavioural genetics is trying to do is to separate out the influence of the genes, the shared environment, and the non-shared environment. You have these three major components. So, and those are variation with respect to particular traits. So the trait in this context means anything that you can measure in behavioural terms that varies between individuals. It might be intelligence, it might be how many hours a week you spend watching television, it might be aggression, it might be religiosity, or whatever. I mean, behavioural geneticists have measured all of those things and hundreds of other things as well. So the shared environment, as you might expect, then refers to all those environmental influences that tend to make children more similar to each other if they're raised in the same family. So the socioeconomic status of the parents and the environment, what kind of nutrition they have. I don't know, if they live on the 45th floor of a huge block of apartments or on the first floor, I don't know, all the things that might make children more similar. Okay, non-shared effects then are all those factors that might make children different. Things like one takes drugs, the other doesn't. One had a traumatic experience, they had a car crash, the other didn't, they weren't there, or whatever it might be. Okay, all the things that might possibly make individuals less similar to each other. Now, what a heritability study then tries to do is to calculate the proportion of variance, that's the total inter-individual variance amongst identical twins or non-identical twins that's associated either with the genes or these two types of environment, the shared environment or the non-shared environment. Okay, so it's all about proportions of variance in a specified population. Proportions of variance have to add up to 1 by definition, and so the heritability then is defined as the proportion of variance in a trait in a specified population that you can attribute to the influence of genetic variation. And that's a very technical specified definition that leads actually to quite a lot of confusion, because you can write heritability then as a percentage, and that's often misinterpreted, especially by the media, as if it refers to the proportion of the inheritance of a trait in some individual, when all it represents is the percentage of the total variance in a population. Okay, it's a population statistic. It has no units, it's a proportion of variance. So there are plenty of, okay, that's the basic idea behind, heritability. And there are certainly plenty of points we can discuss in the whole approach of behavioural genetics and so forth. I just want to point out two important things this evening for the sake of this lecture. The first thing is, the word heritability now is a bit of a slippery word, unfortunately. Heritability in English used to be a synonym for inheritance. But now, ever since the 1940s, in fact, it's gained this technical meaning, which is just like I just defined it. It refers to the variation in a trait in a population due to genetic variation. That's something quite different. It doesn't tell you, when you talk about somebody having 50% heritability for IQ, intelligence, or something like that, it does not mean an individual is inheriting 50% of their intelligence from their parents. It does not mean that. It's a population statistic, and it's a very different kind of thing. And unfortunately, especially as I say, the media do get this badly wrong, and I don't blame them, because the language is very confusing. And some scientists, in reporting their results, also even use the word with the two different meanings in the same text, and that also leads to some confusion. It's worth pointing out, well, again, this is a simple illustration, again. For example, this is to try and understand a little bit, get our head around this technical meaning of heritability. We notice that most people have 2 legs, and unfortunately some people only have 1 leg, and usually, not always, but usually it's due to the environment. They had some accident and they lost a leg. So the variation in the legginess, then, the variation in the population in legginess is 100% environmental and 0% heritable. Okay. Now that sounds a bit strange, doesn't it, when you say that, because we know that you need a lot of genes to build legs during fetal development and so forth and so on. It's just that in practice, the genomic system responsible for building legs always encounters, generally nearly always encounters, the same environmental influences, and so the system builds 2 legs. But it's just an illustration to try and get our head around the meaning of heritability. If there's no variation in a trait at all, then the heritability is going to be 0. The heritability of having a brain is 0 because there is no variation in the population with respect to that particular trait, as far as I know, anyway. Okay. So, in other words, the point here is that heritability tells us nothing really about the complex interplay of components that we were thinking about just a moment ago in the context of dicey, this actual development of the individual. These are 2 different parallel pathways that tell us sort of different things about genetic variation and so on. The other thing to notice about heritability is its very method of calculation, I think, perpetuates this reification, this understanding of dichotomous views of human beings, because as soon as you carve up different components like things like genetic variation, and then you compare it with various environmental proportions of variance, as soon as you give values to something, it's a big temptation to then somehow think this is a thing, that actually heritability is a thing somehow. And that, of course, is not the case. We're talking about statistical calculations, statistical constructs that by definition have to add up to 1, and so that's just the way it is. It's a population genetic study and so on. Unfortunately, again, I think the communication of it tends to give people the idea that somehow you can carve up an individual into environmental bits and genetic bits, and the answer is you can't do that. There's no way you can do that. Now, of course, quantitative behavioral genetics leads directly into molecular genetics as well, huge fields, which we only just touch on briefly this evening. I guess the best known approach to that in the past 10 years in medicine and also in behavioral genetics has been through genome-wide association studies, where what you're doing here is to capitalize on the presence of all these SNPs, these single nucleotide polymorphisms, these single genetic variations. If you like, they kind of act as markers throughout the genome. And the tie to stubby here is fairly straightforward. You genotype hundreds, thousands, maybe thousands of people for these different SNPs, which you can do quite easily now through gene chip arrays, this kind of technology. And then you see what, if there's a statistically significant correlation between a certain SNP and a certain behavior, or maybe it'll be a medical syndrome. And the SNP, of course, may not be right in a protein encoding gene. It may be in a regulated gene. It may be in a non-regulatory region. It may be right within a protein encoding gene. But you can use that SNP then very often to actually identify the genetic variant that's involved. Now, this approach has been really important in medicine and in terms of identifying a whole range of genetic variants which are correlating with, let's say, with schizophrenia and so forth, with various conditions. And that has been a huge success, I think. Unfortunately, it really hasn't been that informative, I would have to say, in the field of behavioral genetics in terms of behaviors within, if you like, the non-pathological population. Because if you look at something like intelligence, actually there was a paper came out last week in Nature, if you're watching on Nature, which was giving a sort of meta-analysis of many different papers to do with the relationship between genetic variation and intelligence. And any single SNP, any single genetic variant you find only gives you a tiny bit of, you can only attribute a very small amount of the heritability to that particular genetic variant. In fact, in the meta-analysis that came out last week, although they were looking at the correlation with hundreds of genes actually in many different parts of the genome, it still only explained the influence of 4.8% of the total variation in the total variation in the population, many populations, in fact, that they were summarizing. And when you think about it, that's not really very surprising at all. If you come back to Dicey and you think about the development of the brain takes thousands and thousands and thousands of genes to do that, okay, working together in collaboration with all the other mechanisms we were talking about to generate the brain. So it's not surprising at all, I think, if brain function, which I guess you need for intelligence, is going to be slightly influenced in different ways to a very small degree by many, 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 many different genetic variants. And that's the picture that's coming out now in pretty much every single trait, behavioural trait, that has been looked at by behavioural geneticists. So in a sense, it's not really that surprising, and to be honest, maybe not all that interesting, because I'm sure, I certainly would believe that genetics is definitely involved in who we are as people and so forth. It's involved in, to some degree, in educational ability, of course, to some degree in sport, some degree in this, and so forth. But it just means that there are hundreds and hundreds of genetic variants that might make small differences in a population. And that leads us perhaps to ask a question. Could it possibly be there might be a single gene which is associated with a 14-fold higher relative risk of being a criminal? That's an interesting question. What would we do if we discovered such a gene? Well, as it happens, that gene exists and it's been known for some time. And that gene is called STRI. Okay, so if you take all the countries of the world, 131 countries of the world, an average of 93% of the prisoners are males. Okay. Now, I can say this because I'm a male, so it's easy to say this, but we are identified by the STRI gene, which is on our Y chromosome, and if you don't have that, you might be something else, you won't be male. Okay, so, and that's the way it is. So what do we do about that? I mean, we don't actually automatically, therefore, put all males in prison. I'm glad to say most of us manage to get through life without committing crimes that send us to prison. That's good news, and so on. Now, you can't go to the other extreme and say this gene has nothing to do with that social factor at all. That's an interesting discussion. But the point here, of course, is I very much doubt there will ever be another gene discovered which will correlate with a particular human behavior to the degree that the STRI gene does. And so I think that helps us to put all this discussion into context. So are we slaves then to our genes? Well, we notice then that there are no genes for any particular behavior. I've emphasized that our own human identities, our own unique personalities are 100% genetic and 100% environmental. And I actually like this verse from the Old Testament, where the psalmist says, "You created my inmost being, you knit me together in my mother's womb." I rather like that metaphor of knitting, because in a way, development biology is like that. You're sort of knit together to make a product that looks very different from the original wool that you started with. It's sort of knitting, great knitting exercise. We also notice that there is no single gene that causes any particular behavioral trait. But this is the key difference. Genetic variants cause differences in a trait. So genetic variance is all about differences. And that is, I think, it's a very basic insight, but maybe is the most important one here. So what about then genetic determinism? How do we define that? Well, I've got a couple of definitions up here. We're going to define that hard genetic determinism is the thesis that given our particular genomes, our lives are not really up to us and are constrained to follow one particular future. And I guess you'd have to say medical genetics, that some medical genetics would be in that realm. There will be certain things genetically. If you have that, then your future is going to go in one direction unless some therapy comes along which is going to help you, and so on. Soft genetic determinism is then the thesis that given our particular genomes, our lives are more likely to follow one particular future, which you might want to say is not determinism at all, really. But here it's a question of probabilities, of predispositions, and I think for most of the population, this is the sort of area that we find ourselves, where there may be genetic variation in our backgrounds that tend us to lean more in one direction or another. And well, here's a classic example. These two guys, I can't remember the name of the basketball team. They're from the same team in the States anyway. Now you might say, you know, this guy obviously has a bit of an advantage in basketball, I would say. And I suspect, you know, tall people tend to play basketball more because it might be a little easier for them. But there are many tall people who don't play basketball. It doesn't determine their behavior. And there are many shorter people who love playing basketball, and so on. So in other words, you may have, you know, this is a fairly obvious example, but of course there are many, many more subtle examples as well that influence all of our lives. We're talking here about probabilities and so forth. Now finally, I want to just speak a little bit about theology and ask the question, what has theology got to do with genetics? It may not seem very obvious when you first think about that. I actually think there's, well, maybe I should say what this conversation is not about, okay? So the conversation's not about trying to find gaps in our genetic knowledge where we're going to introduce the idea of God somehow. This discredited kind of God of the gaps idea, it's not about that, okay? So put that on one side. It's certainly not putting down science in order to raise up theology in some way. No, it's not about that, okay? What it wants to do, this conversation, is to find a sort of theological theme which might be a good conversation partner with the kind of science we've been thinking about this evening. And what I want to suggest is that the Christian concept of humankind being made in the image of God provides quite an interesting conversation partner with genetics. Sometimes in Latin it's called the imago Dei, the idea of the whole of all of humankind being made in the image of God. This has actually been a very powerful trope, a very powerful idea in the history of many ideas of political systems, medical care, education, justification of human rights, moral values. You see the imago Dei popping up all over the place in the campaign against slavery in the early part of the 18th century. Got my century right, 19th century, sorry. You find this idea coming all the time as a justification for fighting against slavery and so on. So it's a very ancient idea, it's a very ancient Judeo-Christian idea, which has been very powerful in the history of ideas. But what does it actually mean? And for that, we have to go back to an original text here where in Genesis chapter 1, we actually read where this phrase is first found, where God said, let us make Adam, which means humankind, in our image, in our likeness, and let them rule over the fish of the sea and the birds of the air, over the livestock, over all the earth, and over all the creatures that move along the ground. So God created Adam, humankind, in his own image. In the image of God, he created him, male and female. He created them. Now, the question is, what would the first readers of this text have understood by that phrase, image of God? What would that have said to them? And I think an important clue here comes from the many contemporary texts in the ancient Near East of that time surrounding the people of Israel, which speak of the image of God as referring to the king. It's used quite frequently, actually, especially in Egyptian literature, but also in the Mesopotamian literature of the people surrounding the people of Israel at that time. Now, of course, they were polytheists, so they believed in the whole panoply of gods, but it was the king, particularly, they say, who was made in the image of God, and sometimes priests as well, but also talked about as being in the image of God. So, when the first readers of Genesis first read this text, they would have been looking at that and saying, wow, that's really interesting, because now what this text is telling us is that these kingly and priestly roles allocated to the few people were now delegated instead by the one created God to the whole of humanity. So, in other words, there was a spread of this kingly role, as it were, to all people, male and female. He created them. And so here was the one true created God who now cares for the physical and spiritual needs of the whole of humankind. So it's an all-encompassing kind of idea. Now, there are two particular strands in this idea of the human God, of humankind made in the image of God, that I would like to bring into discussion with genetics. There are many more actually, but we just have time. I think we just have time for two. So the first then is to underline the value and status of each human individual, irrespective of their genetic status. I think there was a time when the image of God was interpreted more as a sort of list of characteristics that makes humans different from animals. And you know, that list is important and it's valuable, but I don't think that's really getting to the heart of what these texts are telling us about humankind made in the image of God. And really what these texts are about are talking about this delegated responsibility and status that's given to the whole of humankind as by God's love, really. It's something given, it's a gift, if you like. It's not something that's intrinsic so much to humankind. So therefore, humankind's value lies not so much in this view in a kind of list of intrinsic qualities, but rather in this bestowal of delegated value and role in looking after the earth. And I don't know if you know, that word Adam is a play on the word Adama. Adama in Hebrew is the ground. So here we have Adam made from the Adama, theological idea, not a scientific idea, and therefore is put in charge of the Adama, is put in charge of the ground. Okay, so you have the concept of humanity as the earth keeper, if you like, the one who should be looking after the environment and so forth. And so this is important because what it means is that those who suffer from some genetic handicap, maybe some genetic disease, possibly, are just as much made in the image of God as anybody else. Okay, they have value which is not coming from their own ability to function fully with that image of God, delegated authority, but they are those who in fact will receive care from others. The point here also is that the caregiver and the care receiver are both equally part of that image of God. Giving care, receiving care is part of what being made in the image of God is all about, because it demonstrates that common humanity. The image of God is for all people, irrespective of their beliefs or their race or their background or their ethnicity. All people have equal value, and that's incredibly important. And so this undermines any move to make distinctions between people based on their genetic endowment. And I think that's very important. Now, I just want to give you one illustration of what happens when you kind of lose that framework. You lose a framework that gives real value to people. This is actually an article that came out in a respected journal of ethics, medical ethics, just a few years ago. And you can tell what the article is about by just the title there, After Birth Abortion, Why Should the Baby Live? So what the authors in this paper are saying, well, they're following the utilitarian philosopher Peter Singer here, and they're saying, well, it's really sad, you know, that you can't do prenatal diagnosis for every kind of disease because, as we just heard, there are 16 new mutations in this newborn, and sometimes that will lead to disease. Oh, and that's very bad. So you might suddenly find your newborn baby on day 1 or day 2 has a terrible disease. What are you going to do about it? They said, well, after birth abortion. Well, what do they mean by that? Well, they mean infanticide, okay? That's what they mean. So they're arguing here rationally in a medical journal for infanticide of children aged up maybe to 1 or 2 years, because these are not real persons, okay? It all comes around to how you define a person. They say, well, they have a terrible disease, they can't really think straight, or they have some mental problem. Well, they're not real persons, so we can kill them, okay? That's what the journal article is arguing. The point I want to make here is this is where you go, I think, if you lose that concept of all humankind are valued in the sight of God. This is where it can go. I'm not saying it has to go that way, but this is a kind of direction in which the discussion can go. Now, to do justice to this journal of ethics, medical ethics, they also published many responses to that article afterwards, and the issues that led to quite a big debate, as you might imagine. But I just want to point that out as a direction in which the discussion can go. Once you lose an anchor, when you lose a basis for human value, then the slope downwards can go pretty fast. Finally, I just want to, as a second point here, notice that the image of God entails moral responsibilities and duties, which of course might seem very obvious to us. In the context of its time, of course, it was pretty dramatic, because we're talking about a very structured society where, you know, is really just the elite, the oligarchy, the kings and so on, who made all the decisions, and the people who, just the workers and people out in the fields, really didn't have much say at all about what was going on. The point here is that in terms of the context of Genesis, humankind were given this huge challenge of eating, not eating of this tree, this figurative, symbolic tree of good and evil and so on, and they disobeyed. So the implication is they clearly had free will, they had moral decisions placed before them, they went their own way rather than God's way, which had some terrible consequences and so on that we're still living with today. The point here is they had real moral responsibility, they had real genuine free will, and that comes through very clearly in the text. So genuine responsibility. Okay, so I'll just draw this together now before we have time for questions. I want to say this evening, there's nothing, I think, in contemporary genetics except for some severe pathologies that in any way undermines the idea of personal moral responsibility, of genuine free will. I would want to defend genuine free will that we have. I would want to say also that genes do not influence us as if genes are somehow separate from us. We are our genes, we are our brains, we are unitary people and so forth. So we're not living in a separate space from our genome and from our genetic inheritance and so on. We have real moral responsibility. I want to say also there are no single genes for aggression or for gluttony or for crime or something like that. We do have influences, genetic influences in our lives that we would find it very hard to give a percentage to, but you know, they're there in some way, which is sometimes difficult to know. But most important, perhaps of all, is that we are responsible to each other, we're responsible to God, we're responsible to care for the earth that God has led us to look after and so forth. And that includes, of course, the wise applications of science. So we will, I believe, be called to account for how we care for the earth and care for humanity around us. So with that sobering thought, I will finish, and it's over to you for questions and discussion. Thank you very much for your attention. Thank you.